welcome everybody to the Sight Vision and UniProt proteins webinar, which is going to be presented by me, Kate Warner. I'm one of the curators in UniProt, and Emily Bowler-Barnett, who is one of the other curators at UniProt. I'm going to start the presentation and then Emily will finish. She is the curator who is part of that project that we talked about in the abstract of updating proteins to do with human eye health and development. So as you may or may not know, today is World Sight Day. And so we thought we would do a webinar about one of the many curation efforts at UniProt, which aims to support biomedical research into eye health and treatment by focusing on curating proteins in human and model organisms that have an involvement in eye formation, maintenance, and health. So World Sight Day is coordinated by the International Agency for the Prevention of Blindness, or IAPB. It's an international day of awareness held annually on the second Thursday of October to focus attention on the global issue of eye health. So this year's theme, as you can see all over our slides, is Love Your Eyes, which aims to address the issue of eye health at the country and global level by encouraging people to think about the importance of their own eye health and to get a sight test or recommend others to do the same. And in this way, they hope it will highlight the importance of looking after your own eyes while raising awareness of those around the world who can't have access to vision, because everybody counts. So globally, there is currently 1.1 billion people living with a vision impairment, and hundreds of millions more are having ongoing eye care needs. Vision loss has a major impact both globally and for the individual, with profound implications on all aspects of life, including health, education, employment, quality of life, poverty, and many other sustainable development goals. In addition, vision loss is both a contributor to inequalities and is also an outcome of inequalities with links to socio-economic disadvantage, gender, and age. And according to the IAP Vision Atlas and the Lancet Global Health Commission on Global Eye Health report, 90% of people with vision impairment have cataracts or simply require glasses, which are both highly cost-effective treatments available to and is not available to everyone in the world, unfortunately. The Lancet Global Health Commissions on Global Eye Health has highlighted the importance of research to advance global eye health and improve treatments and identify effective interventions. So here at UniProt, we support biomedical research such as visual health by providing a rich resource of freely available, stable, comprehensive, and accurately annotated protein sequence and functional information. So this includes primary sequences, isoforms and disease variants, physiological protein function, including subcellular location, pathways, reactions, interactions, and involvement in disease, structural information, and we also provide a number of tools such as BLAST and alignment. This is what our homepage looks like. Here are all of our resources, our programmatic access, some search and alignment tools and some help of where to get started. And this is how our sequence data is organized into several resources. But today I'm going to be focusing on UniProt KB, which is the central data hub for UniProt. Containing all the functional and structural information on proteins, including disease-related data. So this is the sort of information that we provide in the UniProt KB database, things such as functional annotation, mutations and disease, sequence features, names, links to specialized database, variants, and sequence isoforms and stable identifiers. There are two types of data sets within the UniProt KB database. We have tremble entries. These are entries that can contain all of this information, but the information has not been reviewed by a curator. So it's generated through automatic annotation. And then we have another data set called SwissProt reviewed entries. And this contains data which has been manually assessed by someone such as Emily or myself. And so it's been verified. In addition, we attribute all of this information to its original source. So for SwissProt entries, we'll tell you where that information comes from. Did it come from a publication, by similarity, by UniRule annotations? So these are rules that have been developed by the curators. And for tremble entries, we have these automatic assertion evidence codes. So this will also tell you where that evidence has come from, including the rule, the automatic annotation rule that was used to generate this information. So how can you find eye-related proteins in UniProt KB? So the most simplest way is to just search for eye at the top of our page in the search bar. This is known as a simple search, so it's a sort of free text normal search, simple search, or you can use a complex text query, which you can actually build using our advanced search. And I'd say that this is the most important. This is the most easiest way to find the information that you're looking for. So at the top of the page, you'll see there's an advanced search drop down, and if you press the drop down button, it will give you something like this at the top. So if you search for eye and just do a simple search, you can see that it will search for eye in all of the entries. That can include everything in the entry, including publications. Oh, sorry, I'll go back one. So what it is, so in the advanced search, this is the field, and you can add lines or remove lines using the delete or add. If you wanted to search for eye throughout all of the entries, you can just type in eye and press search. Here, as an example, I've shown you how you can search for eyes in which the function comment, search for the term eye in the function comment, and it's a function comment, it has to have experimental evidence. So here you can assign what type of evidence you're interested in, to restrict your search even more. You can even add, and here you can add an or as well. So you can also search for retina in the function comment by sequence similarity. So for this search, I'll be searching for entries which either have eye in their function comment or they have retina in their function comment, along with these restrictions. In addition to that, you can search for eye in the pathology and biotech disease section, or in the mutagenesis lines, which is important if you're looking at model organisms. Often it will be held there. And you can restrict by taxonomy. So let's say you're only interested in Homo sapiens, in humans, you can put that there and click search, and all of these constraints will be put into your search. Then once you get the results page, you can further restrict your search using the filters on the left. So you can, if you haven't restricted by organism, you can restrict it by organism here, or you can restrict your search to a certain section of an entry, such as gene name or protein family, etc. And you can restrict by whether it's a SwissProt entry or a tremble entry, so a reviewed entry or an unreviewed entry. So that's a bit of a starting point to how to use UniProt to search for eye-related proteins. So I'm going to start by talking about eye proteins in model organisms, and then Emily's going to talk a bit more about in human entries. So one of the aims in UniProt is to understand the mechanisms and the effects that variants have on a protein function, because this is critical aspect for understanding the diagnosis and treatment of specific diseases such as eye diseases. So through expert curation, UniProt provides an overview of available protein knowledge, including disease-relevant data for human entries with a disease association, and this is to support the development of biomedical research and precision medicine. So to support precision medicine based on genomic data, UniProt provides a freely accessible, centralized resource that both links the genome to the functional proteins it encodes and efficiently collates, standardizes, and organizes the knowledge of their proteins and their functions, so that in that way, humans and machines can understand and interpret and rapidly understand what is known about a certain protein, enabling them to design experiments to further understand human disease at the molecular level. An important aspect of our curation effort is the annotation of proteins from model organisms, as you can see here. So most of our biocuration projects actually focus on the annotation of proteins from model organisms, such as mouse, C. elegans, zebrafish, and Drosophila. And this is because much of our knowledge on hereditary, on development, on physiology, and the underlying cellular and molecular processes is derived from the studies of model organisms. So we hope that by providing information on the role of proteins in particular biological processes in model organisms, we can provide insight into the role of their orthologs or homologs and or biological processes in humans. So currently, if you were to perform a search in UniProt, you'll see that we have over 500,000 SwissProt entries. So these are entries that have been reviewed by a curator at some point that have an eye-related term in their function comment. So for this, I used things like photoreceptor cell, retina, optics, eye, etc. And this is the number that I got back. And 20,000 of these, over 20,000 of these are human entries, with 1,539 human entries containing the term eye, and 289 have the term eye in their disease description. So that means that disease description, that suggests that there are 289 entries that have a protein which causes a disease related to the eye. But what's interesting is that the majority of entries that we have is actually from non-human entries. So over 500,000 of these are actually non-human entries. So these are from our model organisms. And 87,000 of these are metazoans. So they're animal. So they're close, they can be closely related to the to humans. And 139 of these have the term eye in their pathology and biotechnology section. So what this means is 139, that potentially 139 entries have a mutation or they have a null phenotype, which results in a change in the eye development or physiology or the underlying molecular mechanisms for eye development. So visual research, of course, is an excellent example of why model organisms are so important in research. Animal eyes can vary in complexity, ranging from a single photoreceptor cell to elaborate arrays of basic units, which allow image formation in compound eyes of insects or vertebrates. So the evolution of the eye required involvement of several distinct components: photoreceptors, screening pigments, and eyes, which orchestrate their proper temporal and spatial organization. And so an analysis of the genetic and chemical components show that many of the evolutionary process have participated in eye evolution. There's multiple examples of co-option of crystallins, GA protein subunits, and screening pigments, contrast with the conserved roles of optins and sets of transcription factors which govern eye development. So as you can see in this picture, these are transcription factors, and they're showing how these different families of eye-related proteins have been utilized by different metazoan. And what this suggests is, of course, that that the regulatory relationship must have been already established in the ancestral photoreceptor cell. So we thought we'd give you one example of why model organisms are so important, and they're so helpful for designing experiments on researching eye health, and how you can use UniProt to do this, to already carry out some of your analysis before you even start your experiments. So this is ATOH7, which is in humans and many other animals. It's a transcription factor that positively regulates retinal ganglion cell fate and formation of the optic nerve and retro hypothalamic tract. The protein actually causes the disease, and this disease is caused by variants. The disease is called persistent hyperplastic primary vitreous autosomal recessive, so PHPVAR. And this is a very bad developmental eye malformation, and it causes things like cataracts and glaucoma and retinal non-attachment. And here you can see we have some information on the variants with evidence codes and cross-references to DSNP, Ensemble and ClinVar, that you can follow to find out more information on these variants. When you run a blast on this particular protein in UniProt, and then align all the 80 and align all of the proteins that have high sequence similarity, you can see that there's quite high. So this is the results of I'll start again. This is the results of an alignment. You can see this is an alignment that's been done in UniProt. And here on the highlight at the left, I've clicked similarity to show which bits of the protein are similar and which bits are very variable. And you can see there's quite a lot of conservation. So this is at the top, there's zebrafish, human, and then you've got mouse, chicken, and frog. I think that is. You can see that for consistency, we try to be as consistent as possible in our curation. So a lot of this, if we think the protein has the same function and is an ortholog, we will keep the same ID, the same protein recommended name, etc., to make it easier for people to find their proteins of interest. So anyway, as you can see, there's quite a high degree of conservation. And in one of these organisms is zebrafish, and zebrafish is a very important model organism for researching for visual research. So this is human eye development at the top and zebrafish development at the bottom. So zebrafish is a very important model organism for research into eye development and disease, because ocular development in zebrafish closely resembles that in humans. And in addition to having the model organism attributes that we need, so they have a short life cycle, they have not specialist living requirements, and they're cheap to maintain and easy to genetically manipulate. They also have a certain behavior that's very interesting for assaying visual function. So zebrafish, after their skin pigmentation, they've changed their skin pigmentation according to light intensity. So if a fish has impaired vision, it perceives itself to be in an environment with low light intensity, and it therefore appears hyperpigmented. So yeah, it's a great model if you're ever looking into visual research. So this is the entry for zebrafish that you saw in that alignment, and you can see it has the same function as a transcription factor involved in retinal ganglion cells, and it has some experimental evidence here as well. And so you can see that there's a high degree of conservation, which suggests that zebrafish might, if you were looking into experimenting with and finding out the function of this protein in human and what parts of the and looking further into this disease, you might consider zebrafish as your model organism because it's so well conserved. And on this alignment, I've highlighted the variant and the mutagenesis sites. So this is human. Here, you can see it's highlighted the variants and a mutagenesis that's been carried out. And if you zoom in, you can see that in zebrafish, it's very highly conserved, which suggests that actually zebrafish would be an excellent model for looking into this particular disease. So I hope that's highlighted how important not just the human entries, but the model organism entries are for looking at animal vision research, for looking at eye development and eye disease. And I hope I've demonstrated how you can use the different tools as well in UniProt to do a little bit of analysis before you choose your model organism or decide which choose your model organism or design your experiments. I'm now going to pass on to Emily, and she's going to tell you a bit more about the curation project that I mentioned at the beginning. Thank you.
Thank you, Kate. So I'm just going to steal Kate's screen. So as Kate said at the start, I'm more of a mammalian curator at UniProt. My background is in human protein signaling, but I also curate mammalian orthologs such as mouse and rat and other model organisms if they pertain to the human protein's function. And today I'd like to give you an overview of how we look at vision proteins, and specifically vision proteins involved in disease in humans, and how they're shown and presented in UniProt, and a couple of the features we have that you can use to explore that. So for those of you with varied biology background, this is the human eye. The vast majority of us are born with them. It's a camera type eye in the sense that it's very much like your camera at home. It focuses light through the lens onto the fovea at the back of your eye, and that is then interpreted by photoreceptor cells that are in multiple layers, such as these, and ultimately transmitted to the brain, where your brain makes sense of them and gives you an image, much like your camera does on your phone. Currently, there are 138 human proteins with the keyword vision. So this is usually when a protein has a role in vision or a function in vision, specifically to seeing light, observing light, or maybe even translating that image in the brain. There are over 2,500 proteins that are mammalian with this keyword. So again, we've got this importance of model organisms and experimental models informing the human protein equivalents, or maybe even going further than that, as these numbers would suggest. So we've currently got several focusing curation projects at hand, but one of them is the human eye, and that's supported by the National Eye Institute. Currently, we have 141 proteins of interest on this target list. We're obviously adding to that whenever we stumble across one or if it's ever suggested to us. And 62 of these have been fully curated as of today. I would like to point out that that's just the human protein. So if I tend to update a human protein or the mammalian orthologs or any other data that's found in model organisms will also be updated. So that's not just 62 entries, that's 62 human entries plus all associated entries with that bit of research, that paper, that protein or function. So I'd like to point out that if you have any site-related proteins or proteins in general that you think need an update, please let us know at help@uniprot.org. We're always able to help and happy to add a protein to our target lists. So I'm going to talk a little bit about a really common disease in the human eye, which is known as cataracts. 43 million people around the world are completely blind, and over half of this is actually attributed to cataracts. And it's really sad that cataracts is essentially a really simple disease in the fact of how it obstructs your vision. And there's a pretty quick fix if you have access to the healthcare for this. So what a cataract is essentially when the light passes through your lens focuses its light onto the back of your eye, and ultimately it culminates in you seeing an image. And this is just how you're seeing this presentation today. But if your lens turns opaque because you've got disordered refraction of light coming back from your lens like this, you'll see the light is refracted back from the lens, which means that it can't get to the back of your eye. So you can't interpret that image as clearly, or maybe not even at all if you've got a severe cataract there. And cataracts, especially in people that have access to decent healthcare, are easily treated with a 10-minute surgery. I've got relatives that have been treated by this, and apart from the idea that eye surgery really freaks me out, it's a really simple procedure that takes 10 minutes on a local anesthetic and essentially replaces the cloudy lens where the cataract is with an intraocular lens that your own eye can use to focus light onto the back of the retina. So this is what a cataract looks like, and this is what one of the lenses looks like that they insert into your eye instead. So I'm going to give you an example of a recently curated vision protein. So BFSP1, also known as phelenzin. This is what phelenzin, the top of the page, looks like in the UniProt website at the moment. So you'll have a short bit of functional information telling you basically the role that this protein has. It's a reviewed protein, so this is in SwissProt, which means it's been manually curated, in this case by myself, and other links on the side that we'll talk about later. Phelenzin, or BFSP1, is involved in lens intermediate filament formation, also known as beaded filaments, depending which bit of research you're reading. And essentially what these filaments do is they maintain the organization within your lens cells. And because of the role of lens cells in your eyes, they have to be incredibly well differentiated, and they tend to form elongated structures. And it's when this organization is disrupted, you end up with cataracts. So this is what the phelenzin entry looks like. Currently, it's got a couple of function comments in here, and this will obviously be added to as the research comes out and other related proteins are curated, and any other functional information can be obtained from there. Phelenzin is expressed, this is the human entry, so in humans it's expressed in the cortex and the retinal lens, so it's quite specific. But if you look across all of the mammalian entries, they're all specifically expressed in retinal lens. And it also expressed throughout development in the retinal lens as well. So it's highly specific to lens development and lens function. So in the interaction section of our pages, it also shows that phelenzin is part of complex required for this intermediate filament formation, and it partners with another family member, BFSP2, and CRY-A, which is crystalline alpha chain alpha, I believe. So crystallines are obviously a key component of the retinal lens, and it's your crystallines that mean your lens is partially the structure it is and why it's so clear normally and you can see through it. So we're in the process of updating these proteins as well. So more information will be added as partner interactions get added to that entry as well. So a little bit of the biology here. On the right, you can see this is what a retinal lens looks like. This is actually a mouse model, because obviously researchers don't tend to cut eyes out of perfectly healthy humans. But these show you the retinal lens cells, and they're all very nicely ordered and organized, and they have this very nice stripy appearance. However, when you knock out phelenzin, you lose this organization completely, and you have a lot of disordered protein formation. And instead of a horizontal kind of striated appearance, you end up with a more vertical, disordered zigzag going off, and this causes opacity in the lens. And essentially, you lose the elongation of the cells in your lens, which means that the light that goes through your lens is refracted instead of passing through. So this is what causes a cataract, and this is how cataract is shown in the human entry for lensing in the pathology and biotech section, and that it's involved in cataract 33, and that this gene causes this disease when it's destabilized or knocked out or there's an issue with it, along with the variants involved. So in this case, we've got a variant at position 348 that causes this disease. That is from a paper publication. So that's been manually curated and added in, and it's also got a link out to DB-SNP. So this is a SNP that's seen in the population as well. And then there's also mutagenesis information that shows experimentally when people have taken a construct or a cell line and they've mutated these residues, what effect does that have on the protein and its function, or maybe even its localization? And in this case, it affects its cleavage by a caspase, and its cleavage by a caspase affects its binding to its interaction partners, and therefore its function in this scaffold that enables the organization of the retinal lens. So I'm briefly going to switch to a live demo now, because I want to show you how you can explore these proteins a bit more graphically on the website. So this is our website. This is the feature viewer. So if you're on the normal entry page, which I showed you earlier, you can navigate by the toolbar down the side here to every section you're interested in. All the popular publications that are curated into this entry are also listed, and we have a feature view, which I'm going to explain now. And the feature viewer is essentially a graphical representation of the protein. So along the top, we have the residue number, 1 in this case to 665. And the graphical representation shows you all the features that are mapped to this amino acid sequence. So you can see at the top, we have domains and sites. And if you click on them, they expand. So you see we've got a disordered region here. We've got PTMs. So an acetyl-alanine is here. And that's imported, that looks like the mouse homolog there. So that's imported from a piece of mouse data in the mouse ortholog. And then there's also oversequence information and mutagenesis data. So as you see here, someone's mutated this residue here, and that abolishes your cleavage by caspase that I mentioned in the slides briefly. And it shows you also that this is imported or curated from its evidence source, and in this case it's a publication, and it's PubMed and Europe PMC links. The proteomics track for those of you interested will highlight all the peptides that are involved in this protein. So if you were to run a mass spec experiment, what peptides are typically seen, so that I highlight the region on there and any evidence and import for that. If you're looking at human entry, it will document any antigenic sequences for antibody binding. So if you're looking to look at this again in experiments in the lab, you might find that useful. And then with regards to disease, we have a variant feature table. So this is a completely unfiltered view, and you can filter these diseases or variants by consequence. So in this case, you can filter just by likely disease, and in which case these variants are typically looked into more and show more significant variants associated with this disease. Or you can search by predicted consequence, whether they're even benign variants. I think we've got any benign variants in here. And variants that are uncertain as well. So the feature viewer will show you all the variants associated with this protein and this protein sequence, including anything imported or manually curated. And they all have information drop-down boxes about the variant position, the variant change, population frequencies, if we've got that information from our imports, genomic location, consequence, and if it's involved in a disease, a brief description and the reference for that as well. And even if it's imported or benign, tolerated, depending on the variant and the data we've got for that. So, because I've clicked that, 3D structure is zoomed in. So the other feature we have at the moment, which obviously has helped with the integration of AlphaFold, is we have a 3D structure of the vast majority of our proteins, especially our Swiss prot proteins in the database. You can navigate yourself around these structures if you want, but equally, if you click on any of the features in the above table, it will zoom in to the 3D structure and show you exactly where that is in relation to the structure of the protein. So I hope that kind of highlights a few of the features we have available for you to explore. Human side of proteins and their disease, with an example of a protein that's involved in vision and eye disease, and also how you can look at orthologs and do your research regarding those as well, and how that might pertain to your interests. So I'd just like to mention our funding bodies, and obviously our target list is supported by the National Eye Institute, but we're supported by a great many other funding bodies as well. This is our UniProt team. We are many and all across 3 sites around the world, and they do a fantastic job, or I'd like to think so. And if you want further help and documentation, we've got a set of training courses up on the EMBL-EBI website. You can always contact us at help@uniprot.org, or you can visit our YouTube channel as well. And just a quick mention for a few things that are up and coming at the moment. We are going to have a new website that's launching in the next few weeks. The feature viewer I've just mentioned will also be integrated into protein entry pages. So the function part of your protein entry page will also have almost like a mini feature viewer to show you where that is, or where the features are in relation to a protein sequence. We've got a new website walkthrough webinar coming up on the 28th of October that Kate is running. So please head over there and register if you're interested in having a walk through our new website as it goes live. And a little bit further in the long term, if you're interested in big data imports and large scale mass spec data in particular, we've got a project called PTM Exchange, where we're importing proteomics, PTM, peptides and peptidoforms into the database, and they will eventually be available with all associated information and links in the feature viewer and protein entry pages as well. I'd like to say again, thank you very much for listening. I hope it's been useful and given you an idea of how you can look at your proteins of interest in UniProt, and how you can do a bit of research pertaining to disease or mammalian model organism morphology. And we'd like to answer your questions, so please get in the Q&A box. Thank you very much.
Thanks, Emily. Can I just nick the screen again? Is that okay?
Yeah.
To answer one of the questions. Right, I'm just going to steal the screen. Yeah. So this just shows the depths of our curators, how good they are. One of our curators at SIB pointed out that those numbers aren't correct that I put on the, and she was right. Elizabeth from SIB pointed out the numbers on this slide weren't actually correct, that there's actually 13,000. She pointed out that I'd put 54,000, which is the number of Swiss entries that are in. So I double checked, and yes, she was right. So actually there's 13,000, over 30,000 entries that have an eye-related term. And most of these are still from animal, just to make that clear, because yeah, that was a really silly thing to do, but there you go. And that answers one of the Q&A questions, which was is the curated info used as it appears in publications, or is it further filtered in order to be part of UniProt? All of our curators have experience in research and writing papers and things like that. So actually we look through all publications and analyze it ourselves, and we determine if we think that the evidence is strong enough to make that function comment or subcellular location. So it's further filtered. Yeah, we're all biologists that have a high degree of research experience. And also we all have a varying range. So for example, if I'm looking at proteomics paper, I'll go and talk to Emily because she's the expert in that. No one comes to talk to me, but unless it's plants
talk to you.
or flies, and then they do come and talk to me. But yeah, we filter it further. We don't put it as is in the publication. We make sure that the evidence is good and we believe it.
I'd just like to jump in there, actually, because Kate does a lot of model organisms and everything's slightly different in how we curate things. And when I curate human entries, we obviously get a lot of disease information in, especially regarding things done in cancer cells and things like that, and tumours. And I'd like to point out that although we do capture as much of this information as we're sure is true data and true evidence, we do make sure that things that only go into function comments and other piece of information like interactions and subcellular localizations, and especially tissue expression, are all very much with regards to the wild type function of the protein. So if all the data in a paper pertains to, for example, cancer and a protein's role in maybe cancer cell cycle, and it's all done with either overexpression constructs or in a cancer cell line, then that data would not be added. If there's any other data in the paper that pertains all to wild type information from wild type models, we add that, and we would probably add a little note in the miscellaneous or a disease comment if we felt we were able to properly explain and be sure that that function was there. But we try and keep the function as much as possible to the wild type endogenous protein function, localization, expression, things like that.
But we always provide cross-references and outlinks to other databases which, may link diseases and things like that to that protein, because they're the experts. So they, that's the importance of cross-references in UniProt really help. But yeah, we only deal with the physiological function of the protein, not like you said, cancer cells and things like that. I think you had a few other live. So
yes, I did. So I had one about variants.
So I'll ask you it. So you feel like you're being asked it. Are the UniProt variants coming from databases that collect these mutations from people all over the world, or just specific regions such as North America or Europe? That's a good question.
So they're from all over the world, essentially. So before I worked at UniProt, I also did a bit of work for the GWAS catalogue. So I did actually do some ancestry and ethnicity information. So I understand how important looking at variants in different ethnic populations and different regions is. We tend to take stuff on, because we curate papers manually, we tend to take it on a very paper-centric view. Currently, we're not curating information with regards to ancestry and ethnicity unless it specifically pertains to the disease or a particular context that it's in which case we'll add a note to the variant. Or even if OMIM have got that linked to an ancestry, that will be added via the import of that. But we tend to collect from databases that take information from people all over the world with any sort of data density across ancestries and ethnicities. And although, I think we're quite aware that some of this is still incredibly biased towards certain ethnicities and things like that, we tend to take it at face value at the moment.
Yeah, and that's also to do with publications, because we need experimental evidence to verify our variants and things like that. So, of course, a lot of publications aren't based upon certain regions of the world, which is a shame, and it's something that I think scientists are trying to address and improve. Definitely. Someone wanted me to put the educational address, so I'll put it back.
I'll steal your screen back and I'll just stick it on.
Yes.
I'll stick. Well, you know what? You can just
Oh yeah, you've got it. Yeah, sorry. Yeah, you stick.
No, it's fine. Had a technology.
Yours is easier.
Before we went live, I do apologise. So I've shorted the training course URL with Bitly. So the EMBL-EBI have got a training website where all the documentation for resources that are hosted there is there, but that is the link to the UniProt specific site. This webinar that is being recorded will go up on there. Everything we've previously done is already on there, and we also have training courses and pages that you can work through that also link in some of the videos from our webinar as well. I like to say we've got this new website coming out, and everything we've shown you today is obviously in the new website. It's all in the same sections in the website. It's just going to look a little bit different. The graphics look a little bit like this on the left, but it should still feel familiar to you, and you should still be able to navigate the same way. we've still got toolbars everywhere. We've still got all our tools at the top and our help links at the top. The webinar for that is on the 28th of October. For the question that someone put in about how to actually get started with UniProt, we've got a webinar on our training courses that is essentially, I think you know the title, Kate. It's
yeah, I think it's a guide for students or something like that by Hamer Bayaji. And it's a really good introduction of how to use UniProt. And actually, I can't remember the exact date, but she'll be doing another one on the new website too. I think it's 2 weeks after my webinar. But search and blast and everything will still work the same. So yeah, I'd suggest going to the UniProt training page and looking at the student guide to getting started with UniProt. I think that's what it's called, something like that. But that's the best place to start. And of course, our help section is very good as well.
Yeah, everything we do is documented, so documented. So everything is on there, and then have a read. And I say there's always, if you really get stuck, you can always send us an email as well.
Yeah, we're happy to help, either for anything, just simple things through to programmatic access and things like that. We love to help our users out and to help them make the most out of UniProt as well.
For those of you looking for programmatic access, we've actually got a pure Python webinar coming up in November as well. So along with all the documentation around that's currently being rehashed. So if you're interested in any future training courses, and especially with things changing on the website, please follow us on Twitter and on Facebook and things like that. And we're always available in the EMBL-EBI training course page as well.